Welcome back, everyone, to Microbiology. In this lecture, we will cover Chapter 8, Part 1, Microbial Metabolism. The objectives in this chapter are to understand the process of catabolism and anabolism, to be able to define and explain the mechanisms of enzymes, understand the changes in free energy during a chemical reaction and the role of activation energy in chemical reactions, to be able to explain the role of environmental conditions on enzymes and how their enzymatic activity is affected, describe the mechanism and role of enzymatic inhibitors in the cell, and be able to use metabolic classifications to describe an organism. Although we tend to look at the basic structures of microbes as simple, their metabolism is incredibly complex. Metabolism pertains to the chemical processes that build up and break down nutrients or structures in a cell. Prokaryotes as a whole have a wide array of metabolic activities. They can live on nutrients that eukaryotes are generally incapable of metabolizing, such as cellulose, petroleum, sulfur, and ammonia. Metabolism refers to the chemical reactions that are responsible for both building up and breaking down nutrients within cells, and it is made of two components: metabolism, which is the process that breaks down complex molecules into simpler ones that usually releases energy. On the other hand, anabolism builds up complex molecules from simpler ones and typically requires energy consumption. Here is a visual representation of this process. ATP represents the energy that is either produced or consumed during anabolic or catabolic reactions. To aid in retention, consider cats. As mischievous little beings, cats represent catabolic reactions that degrade complex molecules into simpler ones. The energy liberated from these chemical bonds is captured into ATP. In contrast, anabolic steroids are used to foster muscle growth. So these substances stimulate the body to create more complex molecules, and that requires the consumption of ATP. Checkpoint 1. Pictured to the right, the metabolic step of converting one glyceraldehyde 3-phosphate molecule into one pyruvate is an example of catabolism. Is this true or false, and why? Similar to a battery, ATP can be charged or discharged. When ATP is hydrolyzed, which means one of its phosphate groups is cleaved off, releasing energy that can be utilized for cellular work, the remaining molecule, ADP, can be thought of as a partially discharged battery. The opposite process occurs during catabolism, where free energy is put back into ADP and is recharged by the formation of ATP. Now, ADP stands for adenosine triphosphate, which means that it has a adenosine group, which is what we see here, and then it has three phosphate groups, and that is the letter A, T, and P. Adenosine diphosphate simply means one phosphate is removed, and so there are only two phosphates, 1, 2. Metabolism oxidizes molecules to mine their chemical energy. In this particular example, glucose is oxidized to produce CO2 and H2O, which are formed by combining the carbon and hydrogen molecules found in glucose with oxygen, a process we will discuss in detail later. This process releases free energy that is utilized to generate ATP. Anabolism, on the other hand, uses the ATP produced during catabolism to synthesize more complex molecules. In this particular example, amino acids are used in anabolism to synthesize proteins. As you will recall from prior coursework, amino acids are the building blocks of proteins. Sometimes you may see them called monomers, which is essentially the building blocks. And a protein would be a polymer, poly meaning many, so made up of many of those monomer units. These two processes are interconnected, forming a cyclic pathway where energy is continuously cycled between catabolism and anabolism. Metabolism involves the chemical reactions that break down and synthesize cellular nutrients, which are regulated by collision theory. Collision theory states that chemical reaction rates are dictated by the factors of speed of the molecules, the energy of the molecules, and orientation that affects the likelihood of chemical reactions taking place. So you can see in this example here, some of these molecules bounce off of each other, but they're not orientated in the right direction. It's only when they're facing each other correctly do those bonds form. In addition, if they don't have enough speed or enough energy, a bond might not form either. We'll talk more about the energy in a moment here. The reactions enzymes assist in are capable of occurring naturally without enzymes, but enzymes accelerate biological reactions by creating an optimal environment for substrates to react in. Enzymes, which are complex proteins, are made up of chains of amino acids that are intricately folded into unique three-dimensional shapes, much like the art of origami, allowing them to serve as efficient tools for catalyzing biological reactions. Cofactors and coenzymes are non-protein molecules that assist enzymes or substances that assist enzymes in catabolizing biochemical reactions. Cofactors are typically inorganic molecules or chemicals, such as metal ions, which aid in enzyme stability and catalysis. Here we see zinc and magnesium as examples. Coenzymes are organic molecules, such as vitamins or nucleotides, such as NAD, which transport chemical groups between enzymes and facilitate enzymatic reactions. We can see this example how a protein may help function to accelerate a reaction. So again, we need molecules to come together in the right orientation. And in this example, we see an active site, which is where our molecule binds here. This is actually a disaccharide that fits in here, glucose and fructose, um, which is sucrose put together. And that H2O locks in there perfectly and allows them to be that bond to be broken between the two. When you take all of the different components together, which is the enzymes itself, any cofactor or any coenzyme that is required, that is called the holoenzyme. And so that's when everything is put together. And enzymes basically do almost everything in the cell, and they're very specific. So they have a high degree of fidelity. So this enzyme right here, it probably has a sole task of only breaking up a sucrose molecule into a fructose and a glucose. And this would be an example of catabolism. And energy is maybe released from this reaction. So we know that enzymes create an environment that is more favorable to reactions occurring. The nature of chemicals is that they want to have less free energy stored in their bonds. The more energy that's stored in their bonds, the more unstable they are. However, some chemical reactions require a spark to initiate the process. Even though they might become more stable products later on, you need something to get the ball rolling. This is called activation energy. And we can see activation represented in this graph is E sub E. In this figure of speech, what we mean is that it takes a little bit of energy to get the process started. A great example of this is the burning of wood. The bonds in wood are greatly made up of carbon-hydrogen bonds. When the wood is burned, this makes carbon-oxygen bonds in carbon, such as carbon dioxide, which store less energy in them. The consequence is there is a lot of energy lost from the chemical bonds in the formation of heat. While this reaction leads to more stable byproducts, it needs some heat, the activation energy, in order to initiate the process. To better understand this concept, we can draw what's called a reaction coordinate, as seen on the left here. The reaction coordinate tracks the process of a reaction. So we can see in the bottom, here's the progress of a reaction in relation to the energy in the chemicals, right here, free energy. And it includes the reactants, a transition state of reactants, and products. The reaction coordinate is a wonderful way to depict this process. Note on the y-axis is free energy. So the higher the free energy, um, the higher um, the higher the compound you find in this graph means the more free energy it has and the more unstable it is. If you examine the coordinate to the right, we have the reactants to the left, the products at the lower right here, and they're more stable. Okay, so this reaction wants to occur, but it does need a spark, so to speak, to get things going, some activation energy. Okay, so, if we go back to our carbon example, the campfire example, the example of wood would be the reactants, and carbon dioxide would be the products. The line demonstrates how much free energy is in the system, called G, or Gibbs free energy. As we can see in the middle, an amount of energy must be added to the chemicals to initiate the reaction. This is a process of getting over this hill and create these intermediate transition states. Okay. In order to get there, however, the chemicals need to go uphill first, so we must put input energy. In the campfire example, we add heat to begin the process, and eventually the heat given off from its reaction can convert more reactants to products. Many times in chemical reactions in the cell, this might be ATP that is used. Activation energy can act as a roadblock for reactions to happen within a timely manner within the cell. So, if there's a lot of activation that's needed, chemicals might not bump into each other with enough speed in order for there to be enough energy to cause this reaction to take place. Um, so you need enough energy so it sticks and this reaction occurs. Some reactions could take decades before they would occur on their own. Consequently, life has evolved enzymes to assist in the process. The reaction coordinate shows how enzymes reduce activation energy by stabilizing the intermediate step of a reaction, leading to the same change in free energy as without an enzyme, but a faster reaction because it's more likely to occur because there's less activation energy required. Stabilizing the transition state allows the reaction to require less activation energy. And this and only this is the reason that enzymes speed up reactions. Therefore, enzymes allow the cell to selectively execute reactions and regulate energy processing. Checkpoint 2. Fill in the blanks. Enzymes make chemical reactions faster by blank, their, blank, blank. Enzymes have a specific process to carry out their function. Initially, substrates bind to the active site of the enzyme, which is the region of an enzyme where substrate molecules bind and undergo a chemical reaction. Then the enzyme catalyzes the conversion of the substrate into new products. Once the product is formed, it is released from the enzyme, enabling the enzyme to catalyze the conversion of more substrates. Hence, the process of an enzyme activity involves substrate binding, transformation, product release, and repeated cycles of this process. In this cycle, note that the enzyme is not destroyed in the process and can be reused by the cell. In the previous chapter, we learned that industrial microbiology is a huge and booming industry, even larger than the pharmaceutical industry in terms of annual revenue. One way companies make money is by producing enzymes that can be used to make products on a large scale. Our Zeta is a company that specializes in designing new enzymes and microbes using advanced computer programs and lab techniques. They focus on converting a plant based chemical called levulinic acid, which acts as a monomer, into other useful chemicals or polymeric substances using a special enzyme they created and improved. This process makes it easier and less expensive to turn levulinic acid into things like solvents, plastics, and medicine ingredients, which allows for more use of renewable materials in various products. Enzymatic activity refers to the ability of an enzyme to catalyze a specific biochemical reaction, and its rate is the speed at which it does so. So, how many substrates does it convert in a given amount of time? Enzymes can be affected by various factors, including temperature, pH, and substrate concentration. Activity of enzymes is generally increased with increasing temperature, up to a point. Beyond a certain threshold, the enzymes can be denatured, which means their three-dimensional structure is altered from its natural state and consequently become inactive. The shape of the enzymatic curve for temperature shows an increase in activity as temperature is increased until an optimal temperature is reached. This optimal temperature, in this graph specifically, lies somewhere around 37 degrees Celsius. So this is probably an enzyme that belongs either to a warm-blooded animal or maybe a bacterium, a microbe that lives or coexists with that bacteria, with that animal. And thereafter, what we're going to see is a sharp decline here in productivity or enzymatic activity from this enzyme due to the fact that this enzyme is now being denatured. And once it's been reached to a certain temperature, it's fully denatured and there's no activity at all. The activity of enzymes is also affected by the acidity or alkalinity of their environment, or pH. Each enzyme has an optimal pH range in which it is most active. Deviations from this optimal range can result in a decrease in enzyme activity. The shape of the enzymatic curve for pH would typically show a peak at the optimal pH with decreasing activity on either side of this range. Enzymes can also be denatured by too high or too low of a pH. Lastly, the activity of enzymes can be affected by the concentration of the substrate that they act upon. At low substrate concentrations, the activity of the enzyme will be limited by the availability of the substrate. As the substrate concentration increases, the activity of the enzyme will also increase, but only up to a point. Imagine the enzyme like a mini factory. As more raw materials are brought in, the more products can be made. But at a point, there's going to be a limit when the factory is acting at maximum capacity and the machines are all busy. Beyond a certain point, the enzyme will become saturated with substrate, and its activity will level off, not decrease. The shape of the enzymatic curve for substrate concentration would typically show a rapid increase in activity as the substrate concentration is increased, followed by a plateau as the enzyme becomes more saturated. Checkpoint 3. What are the three conditions that affect the activity of all enzymes? At times, there can be a wrench that's thrown into the enzymatic machine. There exists inhibitors that interfere with enzymatic activity. They can do so in two main ways: competitive or non competitive inhibition. Competitive inhibition is when these inhibitors directly block the substrate from gaining access to the active site. Since access is blocked, the enzyme cannot perform its function and has to compete for access to the site. However, an inhibitor does not always need to directly block the active site in order to inhibit the enzyme's function. There exists what is called an allosteric site. These are sites that are located elsewhere than the active site that when a non-competitive inhibitor reacts with it, causes the active site to alter its function and decreases the activity of the enzyme. This is very important. Cells create enzymes to orchestrate chemical reactions in the cell. Chemical reactions do not exist in a vacuum, but rather they occur in a chain of other reactions. These are called enzymatic pathways or metabolic pathways, where one enzyme takes a substrate and converts it into a product. The product produced is then converted into another until eventually a desired product is produced. However, the cell needs to be able to turn the orchestra off when it has enough desired product. In this way, the cell is not being wasteful or making too much of a good thing, which could lead to a disastrous consequence. One way the cell does so is through the use of feedback inhibition. This is where one of the intermediate or final products in an enzymatic pathway actually inhibits um, an enzymes earlier in the pathway. This means that the more product there is, the less are made, creating a balanced system. Therefore, they act as inhibitors and may do so as competitive or non-competitive inhibitors by feeding back into the system and causing an interference in this enzyme. This would be enzyme 1 here. This is an example where an allosteric inhibitor works on the enzyme, but you could also have competitive inhibitors that bind right to the active site. Either way, it's possible. Checkpoint 4. Many antibiotics act as inhibitors. What type of inhibition is seen in this animation? We can organize any organism based on the type of sources it can derive its carbon or energy from. Carbon is involved in basically everything inside a cell. It defines what an organic compound is. Carbon can exist in two forms, organic or inorganic carbon. Organic compounds are compounds from living organisms that are made in part with carbon. Carbon in these molecules have carbon-hydrogen bonds. These would include the four basic building blocks of life: amino acids, lipids, nucleotides, and carbohydrates. Inorganic carbon is carbon generally derived abiotically, and so it can be found in minerals, water, and the atmosphere, such as calcium carbonate, sodium bicarbonate, and carbon dioxide. We can organize any organism based on the type of sources of carbon and energy that they, um, based upon the sources in which they get their carbon or they get their energy. Autotrophs and heterotrophs are the categories for what source of carbon the organism harnesses. An autotroph is capable of deriving its source of carbon from inorganic sources and converting it into organic carbon within the cell. An example of this would be plants, algae, or cyanobacteria that are able to capture CO2 from the air and turn it into cellular material. Heterotrophs, such as animals, can only repurpose existing organic carbon, so they must consume other organic materials for survival. The obvious examples being plants for herbivores and animals for strict carnivores. Phototrophs and chemotrophs define the energy source. Phototrophs use the sun as their energy source, again, such as plants, algae, and cyanobacteria. Chemotrophs use chemicals as their energy source, such as animals, fungi, intestinal bacteria, just to name a few. These are all examples of chemotrophs. Combined, we have an organism's metabolic classification. So, for example, a photoautotroph derives its energy from the sun and its carbon from inorganic sources. A chemoautotroph, however, would derive the chemicals from would derive its energy from chemicals and would get its carbon sources from inorganic sources. Another example here would be photoheterotroph. So it means that it gets its energy from the sun and its carbon from organic sources. And that is the end of chapter 8, part 1. Thank you all for being here, and please watch chapter 8, part 2, where we will examine more closely the metabolic pathways used to harness energy. It's going to be a little bit more intense in this chapter, but I felt like it was necessary to break them up here because there's going to be a lot more information that kind of goes in hand in the next part. So please join me for the next one, and thank you all for being here.